Hello biologists, we're going to talk about the structure of RNA today, talk about transcription, and then a little bit about translation. This will help you with quiz 106, 108, and a little bit of 1.12. Before we talk about RNA, we're going to remember a little bit about DNA. Remember, DNA is the instructions for how to cook everything in your cell. It contains all the genes for any cell in your how to make any cell in your body. And the DNA is in your nucleus. When you copy all of the DNA to make more DNA, so you have two copies, like you do when you make a new cell, this is called replication. Let's take a quick look at an animation of replication so we can make some comparisons between DNA and RNA. The link to this animation is in the notes on YouTube. So you can see that DNA has two sides. It's kind of like a zipper that has two sides. And as we start to play the animation here, you can see that an enzyme right here starts to come in and unzip the DNA. And that the DNA starts to be copied by other enzymes here. Let's play a little bit more. You can see that both sides of the DNA are used to make new DNA. Enzymes keep working and eventually you have two whole new strands of DNA. So the process we just looked at was DNA makes DNA. That's replication. What we're going to look at a little bit more is how DNA makes RNA. That's the process of transcription. And if you remember, when RNA that comes from DNA makes protein, that's called translation. Here are some more details about the big ideas. DNA makes DNA, that's replication. When DNA makes RNA, that still happens in the nucleus, that's transcription. And I put a big space here because you can mentally kind of draw in the nuclear envelope because this process takes place outside the nucleus. It's different than replication for making a new cell or DNA to RNA transcribing a single gene. RNA makes protein outside the nucleus, so translation happens outside the nucleus using mRNA, rRNA, and tRNA. Protein, of course, is produced outside the nucleus in the ribosomes. Here's the nucleus of the cell, and outside the nucleus are the ribosomes where protein production goes on. RNA is made from DNA, so it can be transported outside the nucleus to the ribosomes. These are the ribosomes, these little blue dots here. They're on the rough endoplasmic reticulum, and they can also float free in the cytoplasm, but many of them are right here outside the nucleus. So how do proteins, ribosomes, RNA, and DNA all fit together to make proteins? Well, the first step is transcribing the DNA to RNA. We need to get certain information from the DNA. That information is in a single gene at a time. A single gene is transcribed at a time from the DNA in the nucleus out of the nucleus to the ribosomes where that information can be used to make a protein. This process of transcribing, turning DNA into RNA, is called transcription, and it goes on inside the nucleus. Here's a good diagram. Transcription is going on right here. We'll look at a larger picture in an animation in a moment. Transcription is going on here. You make RNA, and then that RNA travels outside the nucleus to be transcribed. Let's look at an animation of transcription and compare it to replication. So you can see that transcription starts with DNA. It starts with the same thing, but let's play this animation here. You can see that an enzyme moves in. Enzymes are very important in all these processes. And again, DNA is opened up, but you can see it isn't opened up all the way. and that you don't get two different complete DNA molecules as you did in replication. You're getting one single-stranded mRNA here. Just the middle of the zipper is opening up, it's making RNA, and then it's closing again. And that the RNA that's produced here in transcription is different in a couple of different ways. It's single-stranded, DNA here is double-stranded, RNA is single-stranded, and some of the bases are different. You can see that the blue here is U in RNA and T in DNA. So those are a couple important things to remember. RNA is single-stranded and it has one different base. It has U instead of T. That's uracil instead of thiamine. DNA is transcribed into RNA, so information in the nucleus here can come out to the rest of the cell. DNA never leaves the nucleus. It stays there so it can be protected by the nuclear envelope from all the other things going on in the cell, but it sends a messenger out to the ribosomes. And that's what transcription is about. It's making that messenger, that RNA, so that it can move out to the ribosomes where protein can be made. Remember, mRNA is like a recipe card. This is DNA. This is the huge instruction book of how to do anything you want in any cell in your body. But this is a clunky thing to have in your kitchen or to work with. So you want to copy or transcribe something into a smaller package, a single recipe on a single recipe card. That's what mRNA is. It's the code for making one protein on one recipe card. And it's carried to the outside the nucleus to the ribosomes. So what kind of DNA is the recipe card? Well, we talked about DNA being a messenger. The recipe card is messenger RNA, or mRNA. Messenger RNA is made in the nucleus and travels to the ribosome. It's made in the nucleus and it's going to travel outside the nucleus to the ribosomes where protein can be made. Let's talk a little bit more about the differences between DNA and RNA quickly, because this is an important part of understanding how they both work. DNA has two strands. You can see there's like two sides of a ladder or a staircase here. It has the bases C, G, A, T. In contrast, RNA only has one side of the ladder or one side of the staircase. It has three of the same bases, but this base here is different. This is uracil, and over here in DNA it's thiamine. So it has different bases. It's single-stranded, and RNA is made in the nucleus, but it works outside the nucleus in the ribosomes, which are in the cytoplasm. So there are three different ways that RNA and DNA differ. RNA is single-stranded, DNA is double-stranded. The easy way to remember the bases is G-cow versus G-cat. That means adenine pairs with uracil in RNA, that G-cow, instead of thiamine in DNA. Adenine and thiamine pair in a DNA molecule. DNA stays in the nucleus. RNA is made in the nucleus, but it travels outside to work in the ribosomes. See if you can pick out a couple important differences in this diagram. Or this diagram. Here's a really simplified picture of RNA. It has one strand. There's no T, there's a U instead. It has the same steps as DNA, but remember, there's no thiamine, there's no T, there's uracil, U instead. DNA needs to move information in the genetic code to the RNA in order to make a protein. You need to make a copy of your recipe of a single gene and move it outside the nucleus so it can be used. This messenger RNA is made from DNA. You can see only part of the zipper is unzipped here, and the mRNA is being made in the middle. The DNA does unwind just a little bit at a time. The RNA is built using DNA as a pattern, and then the DNA zips up again. The new mRNA is only one strand, and it has uracil instead of thiamine. So now mRNA goes to the ribosome. When mRNA goes to the ribosome, that's translation. When it gets outside the cell, or outside the nucleus, outside the nuclear envelope, and it travels to the rough endoplasmic reticulum, or anywhere there's a ribosome, and it starts working with the messenger RNA starts working with tRNA and rRNA to make a protein to translate that recipe into a protein. That's translation. You can see here's a very simplified picture. Here's your messenger RNA. Here's your recipe of one gene, how to make a protein. And it's working with tRNA, which brings in amino acids to make this protein. Remember last time we talked about how proteins are strands of amino acids strung together. They're different amino acids all strung together, and they curl up in particular shapes to do particular things in your body. So mRNA goes to the ribosome to make protein. You can see the small subunit here of the ribosome attaching to the recipe card, the messenger RNA, that's got the code for which amino acids should line up to make which protein. tRNA is another kind of RNA that brings in the different amino acids. It matches the code on the mRNA here and brings in a specific amino acid to attach to the growing chain of amino acids. We're not going to watch an animation of translation yet. We'll do that in another video, but I'll put the URL up in the notes if you want to see it. You can see the structure of tRNA here. It's one of those, it's RNA that's folded up on itself. And it has two important spots here. The codon, which reads the mRNA, and the area where the amino acid attaches here. This is the translation molecule. It's the transfer RNA molecule, and it's kind of folded up on itself. Unlike messenger RNA, remember, which was a straight line, messenger RNA here is a single strand and kind of works best when it's all unfolded. Ribosomal RNA also curls up on itself. It's another type of RNA that crumples up to make the ribosome that helps line everything up, the tRNA, the amino acids, and the messenger RNA. The ribosome here is crumpled up rRNA. So messenger RNA here is the only straight RNA. So let's do a little quiz practice here. Which type of RNA does not curl up on itself and attach to itself? Is it messenger RNA, ribosomal RNA, transfer RNA, double-stranded RNA, or you don't know? Hopefully you remember that messenger RNA stays straight and single-stranded when it's being used. So the correct answer here is mRNA, messenger RNA. What is one difference between RNA and DNA? Well, these are all good answers. DNA is double-stranded, RNA is single-stranded. RNA has uracil, DNA has thiamine. RNA can leave the nucleus, DNA cannot. RNA is shorter because it's only coding for one gene. DNA codes for everything. A whole chromosome is very, very long, and RNA is just one part of that chromosome. So, thanks so much for watching, and go take quiz 106, 108, and read 112.